What is the hypothesis about Acumen's ACU193 and how that might be different in terms of what it offers to patients? How would it be different from the current standard of care?
In terms of how ACU193 is different than other things that have been tested in the past, you have to talk a little bit about what's called the amyloid hypothesis broadly. Now, there is a tendency for some people to think of the amyloid hypothesis as just one thing, but it turns out that you really have to dissect it into its different parts to really understand it. And so some of the drugs that have been tested in the past were really targeting this A beta protein, just the single ones to stop it from being made, for instance, with a couple of different approaches. So that was one approach, is to just try to get rid of the single A beta proteins. The other approach that's been kind of more recent is to use these monoclonal antibodies to get rid of the plaques. And so that really would be amyloid plaques. Um, but there are other species that are in between. There's one called protofibrils that's kind of in between a beta monomer and a plaque. But what ACU193 focuses on is what's called oligomers. And so what that means is if you have a few of these A beta proteins that stick together, there's a lot of evidence that's accrued over the last 20 years or so that that's really the toxic species. And so ACU193 was designed from day one to target that toxic species. And so it is different, even though broadly all these things are affecting some part of either the A beta or the plaque or some part of the cascade. There's been nothing to date that selectively has targeted these oligomers, and that's what makes ACU193 unique.